So now I'd like to give the floor to Max Weigend. Max has been involved with animals and plants since he was a child and has been collecting plants in his own garden since the age of 10. Yeah, after an international career, he became director of the Botanic Garden of Bonn in 2011 and president of the Association of Botanic Gardens in 2016. And he specialized in urticaceae and in loasaceae. Max, the floor is yours for about 20 minutes.
Okay, hello everyone. I'll try to keep to the time. I have been asked to give a keynote on documentation, record keeping, and I will try to share some of my thoughts with you. I haven't participated in earlier meetings, so probably I'm just telling you what you already know, but you know it's always good to give a talk where everybody agrees so we don't have any conflict brewing. Maybe I've got some ideas which are novel or not universally accepted towards the end of my talk. I'll try to share my screen with you. Now, here we are. So my talk is entitled Accumulation versus Collection, Documentation Makes a Difference. And I'm going to first discuss what we are actually dealing with when we document a collection. What's the whole idea? What is a botanical garden? Now, this is kind of a botanical garden. Our colleagues from Hanover might disagree. That is Herrenhausen, which has a part which is a botanical garden, but much of it is actually largely ornamental plantings. And the obvious question to ask is, why is this not a botanical garden, and how is that distinguished from a botanical garden? Now, this is a botanical garden, at least I sincerely hope so, since I've been director of this particular botanical garden for the last 9 years. Now, where's the difference? One difference is, and one crucial difference is documentation. And we'll come to a definition a little bit later. This is a picture from Florence Botanical Garden. You can see all the potted cycads here and some trees here. And if you look at the labels here, the label of a cork oak, then you can see that they've got numbers. And that, of course, refers to the fact that there is some kind of data associated with that plant. And as we all know, the standards expected from plant documentation have been gradually increasing. That is something that I'm trying to visualize with these labels representing 4 different time layers in our botanical garden, starting off with people being perfectly content with having a name on a plant without wanting to know where it's from, to having a number on it, but really only a number in a sense of a catalogue number, like Wallich would have used it in the 19th century, to coming increasingly towards coded numbering, as you can see that on the right-hand side, and an increasingly extensive documentation. Of course, it's a different question whether that's only in the database or whether that's also on the label, but the key message is that our requirements for documentation have gradually increased. And that, of course, has a whole range of different reasons. One of the reasons is that that botanical gardens have an extensive role, for instance, in teaching and outreach. And these are just some pictures from outreach and teaching elements from a range of different botanical gardens here in Brazil, in Germany, in Italy, where you can see that obviously that is also a defining feature of botanical gardens, that we communicate science to students, which we then call teaching, or to the population in general, which we then call outreach or education. And of course, the generally accepted definition of a botanic garden, as contrasted with a public garden or a park, is that Peter Weiss Jackson formulated that in the following: botanic gardens are institutions holding documented collections of living plants for the purposes of scientific research, conservation, display, and education. And in the following few slides, I would like to make a direct connection between the first half and the second half of this sentence, between the documentation and the purposes. If the plants in botanical gardens have specific purposes, which may be overlapping, then we could claim that there are generally five plant families in any botanical gardens. And those are the teachiaceae, the researchiaceae, the displayaceae, the educaceaceae, and the conservaceae. So those are plants which perform a specific role in one or two of these fields. Everything else doesn't actually play a prominent role in a botanical garden in the strict sense of the word. And actually, I grayed out the display AC because display AC are not a defining feature of botanical gardens, unless unlike the other four groups. And the obvious question is, if these are the 4 or 5 plant groups that we have in botanical gardens, which specific reasons do we have for documenting them? Why should they document it, and how should they be documented? That is, which data would we like to store? I think the question is fairly easily answered in the case of the conservacy, the plants that we keep for maintaining them in our gardens for purposes of conservation. Here we are forced to ensure that they survive in the gardens because they can't in nature for whatever reason. But in order to actually meaningfully preserve them, we have to make sure that we actually maintain not only the species, but that we maintain some semblance of the genetic makeup of that species in our ex situ collections. If you look at on the left-hand side, you can see a distribution map of Arnica montana, a quite important medicinal plant in Germany. And you can see that it's got population clusters scattered over the entire country, larger ones in the east, smaller ones in the west. And of course, if you want to preserve the population in its the populations in their genetic diversity, then you actually have to make sure that you preserve those different subpopulations in different places. And Bonn Botanical Garden, for instance, has a program conserving particularly this tiny subcluster of populations close to Bonn city. So we're conserving a marginal population of a widespread species. We have to conserve the local ecotype. Everything else wouldn't make any sense. And that, of course, requires detailed documentation in order to know that it's actually material from that subpopulation and not from the one on the other side of the Rhine or down in the Saarland. So here I think it's fairly obvious. If you look at the ex situ conservation website of Verband Botanischer Gärten, the German Association of Botanical Gardens, then you can see that all the populations conserved in the network are all documented with a code and with detailed locality information. What we are essentially dealing with here is a kind of chain of custody. So we want to have complete traceability of the accessions. Why? Well, if we're talking about endangered species, we have to document that we legally obtained them because they're usually protected under state law or national law. If we want to reintroduce from our ex situ collections into the wild, and we introduce from the wrong subpopulation, we may actually contribute to genetic erosion rather than reducing genetic erosion, which is the main aim of reintroduction into the wild. And of course, if we don't know what exactly we're having, we can't verify whether we are successfully preserving genetic diversity. Even worse, and that sometimes happens, that for simple reasons of ease, ex situ collections are propagated vegetatively, which entirely beats the objective, because then you start cloning the plants and you lose all genetic diversity. So ex situ conservation, I think it's immediately obvious, requires comprehensive documentation and extended record keeping. What about science? And we'll start with something very trivial, Urtica dioica. Few of you have them in their gardens, at least not voluntarily, probably. It's one of the most common agricultural and roadside weeds you can possibly have in Western and Central Europe, actually going right into China, so really widespread. If you look on the right-hand side, you can see that this species has got a whole range of different ecotypes or morphotypes, so they look quite different. And if you start doing phytochemistry, you will also find that from that species complex, Urtica dioica, you get dramatically different chemical profiles. Now, what does that mean? That means that analyzing Urtica dioica and not documenting which specific accession, which specific individual you analyzed, gives you scientific data which you can publish, but which nobody will ever be able to reproduce, because the profile that you obtain from your given clone may not be present in any other clone anywhere in the vast distribution area of that species. If you can't falsify a scientific result, it's no longer science. It's that simple. So you have to record, you have to have traceability. And that's why I use the unusual term chain of custody here. In addition here, having a name in Urtica dioica, even an infraspecific name, is essentially null and void because the infraspecific taxonomy is completely confused. Yeah, so there's no other way apart from direct documentation of the accession to verify that you are able to reproduce scientific results and compare them to those of others. The same thing goes, of course, for molecular data. This is a phylogeny, a truncated phylogeny of Urtica dioica and its allies. Here you've got exactly the same problem. They often look very similar. The misidentifications in herbaria and collections abound. So unless you can go back to the original material in some way, and the most obvious way of having a fully documented accession, you will not be able to reproduce even phylogenetic data for a given question you may want to address. The obvious question to ask is how good are gardens in that respect? So if I want to have only fully documented material, and I want to go to botanical gardens to use that for my research or conservation, then it would be good to know, so what's the percentage of well-documented material in botanical gardens? And we more or less accidentally did a study of that on the genus Corylus. We wanted to do a Corylus, a hazelnut phylogeny, and requested material from botanical gardens, and we got their data. And it turns out that if you look at German botanical gardens and neighboring countries, then we got reports of 219 accessions of Corylus avellana. 100 of them were native common Corylus avellana, and 191 were undocumented, which means 88% of the Corylus avellana, of the Corylus in botanical gardens, and those that answered, is essentially useless for a scientific study. At least if you want to do a comparative scientific study, because you would never know whether the material you get from Hamburg is the same clone as the one from Mainz and where it originally came from. Yeah, so only 12% actually make that litmus test. That is only 12% of the Corylus species, genus that's present in just about all temperate gardens are actually useful in that strict sense of the word because they are sufficiently documented. So what does that mean? Documentation for collection use and research. Research results are not fully reproducible if documentation is incomplete. The same or different genotypes may be present in different collections. We know a lot of clones abound in collections. Phylogeny or phytochemical screening may be unpredictably biased by multiple samples of the same genetic individual from different collections. The representativeness of the data is unclear because we can't correlate that to distribution and populations. And maybe most importantly nowadays, the legality of the research is unclear. Because nowadays you have to be able to prove exactly where your material came from in order to be sure that you were allowed to do the type of research you actually carried out. And that goes back to the Convention on Biological Diversity backfiring on science via the Nagoya Protocol. I think most of you will be familiar with it. Most countries represented here by in this Congress have countersigned the Nagoya Protocol, and it essentially what it means is in order to be allowed to do any study on the genetic or biochemical composition of a genetic resource, you first have to verify when and where exactly was the material sourced. So where was the access done and when? And secondly, where and when, and secondly, which additional rules may apply. So you actually need that information. Otherwise, you can't simply go ahead and do your research. Actually, you can do your research, but you can't publish it subsequently. And in many countries, you actually liable for legal prosecution if you do. In Germany, certainly you are. So one fairly nifty way of dealing with this problem is, of course, IPEN, International Plant Exchange Network. I think most of you know that. It standardizes plant records and the transfer of information. It regulates access and exchange and also transfer of material to IPEN and non-IPEN member, and has ABS provisions. So the source of the germplasm via IPEN remains traceable. We are back to the chain of custody, and it characterizes the legal framework which the resource is under. Yeah, just a reminder. I'm sure you've talked about that before. Now, what type of information has to be retained? This is one of my favorite examples here. Material brought into gardens, and that's relevant to both conservation and science, is nearly never representative of a species or even a population. And this is just an example here from Crook Farm. I think all of you know this fantastic nursery in Wales. And they spell out where their material is from and characterize it. And in that, they are, of course, the rare exception. Usually, if you get commercial material, you may even be told where it's from, but you won't get any qualifying information. And here you can see those are 3 representatives of Cotlea spicata, all given fantasy names by Bledin and Su, and coded with a code number, and then you get a source area. Now, if you lose that source area, the material immediately loses its value, but you can also see that here, for instance, in this single catalog, from the same Darjeeling area, you get different ecotypes or morphotypes of the same form. Reducing them to a species name and giving them a number would, of course, be a major fallacy. So what we need in order to make science, we need this chain of custody. We need a direct connection between, on the one hand, all the codified information, usually transferred via an accession number. We need it to be linked to specific time and date and possibly person of collection. And then we can tie this accession to all the scientific data we generate, to the genomic DNA, to chemical profiles, to sequence data, and to all images and observational data we may have here on the left-hand side. And of course, that accession ideally always or nearly always corresponds here to, for instance, a seed collection or a seed sample. Ideally, that seed sample comes accompanied with a voucher for verification. It may be impossible to make a determination of the seed, but once you've got the plant growing, or you collect the living plant, then you can make a voucher. Why would you want to have a herbarium voucher with a plant collection? Well, in many cases, that's superfluous, but not necessarily. Think of the many plants we have in our collections which change under our care. One common example in German botanical gardens, I think the only commonly found species of Aquilegia is Aquilegia vulgaris. And I think in hardly any botanical garden are you going to find the wild type, because it constantly bastardizes with closely allied either species or cultivars of the same species. So if you want to be able to verify if the species that you've got growing in your garden still is the species that you introduced maybe 20 or 30 years ago, you have to anchor that accession in some way. And of course, the classical way of doing that is by vouchering it in a herbarium specimen, which you can then compare to the collection that you still have growing. Herbarium specimens and increasingly genomic DNA are important to track, the process of domestication that may take place. Here, one of my favorite examples, a Chaeophora on the right-hand side. That's what you find commonly in botanical collections, in botanical gardens. I call it the garden type. It was introduced into German or European botanical gardens about 150 years ago, and looks like the pictures on the right-hand side. The wild type here on the left looks completely different. What has happened is simply that in the course of 140 years, of growing in gardens, it has been completely domesticated. So I think I convinced you that we need this documentation and all the records for science, and we need it for conservation. Now, what's with display AC? The plants that we have for showing. And of course, if you look at the tulips at Keukenhof, you may not want to accession them into your database, because come June, most of them are thrown onto the compost heap, and there's little point in accessioning the new bulbs each year. On the other hand, some plants primarily used for display may be an integral part of the collection, may be actually an emblematic part of the collection, like those Chilean honey palms here in Pisa Botanical Garden. And very often these then have some kind of educational significance. This is an example from our botanical garden here in Bonn University. So these Taxodium, these bald cypress, are historically relevant, and we are fortunate that we know who actually collected them, for whom they were collected. They're part of our garden history, and so that information doesn't give them any conservation value, because there's lots of bald cypress in South Carolina still standing, so they won't come back to us for seeds, but they've got a conservation value. That's not a spelling mistake, actually. So that is an educational value that these plants have, but they only have that because we have the historical information attached. So
You have 3 more minutes.
Yeah, coming back to our scheme, I think that's what we can claim. All these plants, all these plant groups require their documentation. And I think you can read at your leisure what's standing here. So there's a lot of added value and legal requirements involved. So how does record keeping tie in with collection quality? Record keeping, of course, first and foremost, documents collection scope and size, and records taxonomic identity and taxonomic and phylogenetic diversity. And of course, this has to match the garden mission. It defines conservation value, scientific value, documentation of the legal framework which ties into scientific and conservation value, conservation value, and horticultural quality, which I didn't talk about very much, whereas horticultural merit stays a little bit out of scope here. Conversely, what are the particular challenges? I already mentioned that hybridization in gardens is a major challenge. And if you grow, for instance, Pulmonaria or Aquilegia together, you can't actually, it's very difficult to keep track of what you're dealing with, what you have in your collection. And a dramatical problem are outcrossing trees. We've got, for instance, two carrier species here standing side by side, but there wouldn't be any point in sowing the seeds because all of them are going to be hybrids, and I do not know how to reflect that anywhere. An accession should, in theory, be a subset of the genetic phenotypic diversity of a wild population. Here you can see the highly divergent seedlings of wild collected Rus seed. But as a matter of fact, we usually, or very often, do clonal propagation. And accessions may then represent single genotypes. Here, an example from our garden, we brought Eriogonum ovalifolium into the cultivation. You see 3 different clones, and now we are propagating them with cuttings. I've got no idea whether these cuttings represent all 3 genotypes or whether we've only got cuttings of the most vigorous one growing on the lower right. So we're losing, if we don't take care of that and don't keep track of that, we're losing the genetic diversity of that accession. And that may mean that we end up with single clones, and if we're unlucky in dioecious plants, we'll end up with a single gender. Okay, so that brings me to a whole range of challenges in record keeping. And I'll just briefly go through them. How can record keeping help with mission match? So with what we actually want to do with the collection. How can we optimize conversation value? So that's the educational value, and use record keeping for that. I mentioned the historical perspective. How can we optimize conservation value? And that includes questions such as how to track genetic diversity and how to track genetic integrity, excluding hybridization. How can we increase scientific value? Very much the same questions. How can we facilitate timely and complete transfer of the record data? That's a major challenge. As all of us know, it's very difficult to obtain the data from different gardens very often. And very important and increasingly important, how can we increase legal certainty for our collections using the tools at our disposal with record keeping? And with these questions, I'm hopefully on time and I'm definitely finished. Thanks a lot.
I would like to ask Alexandra. Hey, Travis' children are joining. I would like to ask Alexandra to join your most interesting idea.
Yeah, hello everybody. So we had little technical problems because I wasn't allowed to share the Jamboard, and I had to send the link via email to Christina, and it was about 5 minutes until we could start, and we introduced each other. We didn't discuss as long on the questions, but we found that especially for the first point, that there is a collection policy is especially important, and we discussed about documenting the genetic diversity in the collections, and how important it would be to have a garden herbarium, but for my garden, it's too small. The herbarium is too small. It's too much effort to do it also.
Okay. Yeah. So you had the technical problems first, and then you took the time to actually get to know each other. And yeah, and you had a bit of time to discuss what you wanted to.
Yes, but we had the keynote speaker joined our session, so the discussion was very interesting.
Great, thank you for sharing. I'd like to ask room number 2 with Alison.
Hi, yes. Well, I enjoyed trying to use every single color of post-it notes to start. No, it's I did manage to share my screen, and we had quite a long chat about the. We were talking a lot about the conversation value of plants, because that's a lot of the people in our group. We felt that probably the higher percentage of plants in our gardens were going to be of conversation value as opposed to conservation value. We struggled a little bit because of the questions were so wide-ranging and fundamental. It felt like sort of solving every single thing about botanic gardens in 10 minutes. But it was nice to see everyone and nice to be able to chat, and technically it seemed to work pretty well.
Yeah, great, thank you. So let's keep on the conversation. And I'd like to ask room number 3, Åsa, what would you like to add to what has been said already?
So yeah, we could share. Maybe I wrote in a different. Yeah, there we are. No, we were talking a little bit about these things, but as Alison said, quite big questions, right? So in the beginning, I forgot to type everything down that we were thinking of. But then you reminded me, someone in the organization board, and we got some stuff down in the end there, talking about how good record keeping could actually help you see, get a lot of data out of your data, so to say. If you want to do something, if you're celebrating a big anniversary for your garden, for example, see how many trees are of a certain age, or you could report back to your board to see if you have a lot of wild collected specimens and so on. And we also talked quite a bit on how to communicate with the scientific community about the collections. Yeah.
And how would you do that?
We were talking a little bit about what kind of tools the scientific community uses for data, and GBIF came up, and it talked about it's quite easy to upload your data from IRIS to GBIF, and how that can improve your data used in science.
Thank you. Yeah, so now we still have a bit of time, about 3 minutes for other facilitators to share something if they feel that they have something burning they would really like to share with all of us. Be quick, unmute yourself and take your chance.
Sure, Matt's here from Gothenburg, Group 10. We just started by saying that often the display is the main focus of many botanic gardens in Arboreta, we think. So it is a challenge to sort of backtrack what we have, but in many cases data do exist. And if we just connect with each other, the accessions, even if they're not valuable for research and conservation in their own right, they would be in a global context. Like, I mean, we are a meta collection, so I think, and there tools like IRIS can definitely help in creating that community of meta collections. And also just, we've also noted that it's very rare that you have a good herbarium in botanic gardens if they're not from the start part of a university garden. But even just sampling the collection for well, leaves or just small samples to be able to make DNA studies could help in this context if you see it as a meta collection. Yeah, that's about it.
Thank you. Is there anyone who'd like to react on that? Max, I see you.
Just one very brief comment. I mean, it's absolutely impossible to systematically voucher a botanical garden. And we've also got some 10,000 species. That's absolutely inconceivable. But what we do as a matter of course, if that anything is used in a scientific study, anybody sends samples for a specific scientific study, that is the moment when a voucher ends up in our institutional herbariums so that we can trace the sample that was sent out back to something that sits in our permanent collection. We don't have the ambition of vouchering everything that's in the garden, certainly not. But in a tandem, I think that's quite sensible.
Thank you, Max. We have time for one more person feeding work. One more comment. Who is the one? Just unmute yourself because I can't see you all at the same. Abby, I see your hand.
I think our main takeaway from our conversation is really the importance of having so many people on board, from horticulturalists to the people that are collecting your data, and really how do we develop kind of that group effort of getting the best data for our collections.